specific for that receptor. This is a B cell with the immunoglobulin receptor. Binds to the antigen, right? Brings it in, digests it, and puts out a piece of it. Right? Here's the T helper. All right. So now what we've gotten is we've got a B cell that found an antigen, right? Bound to it. Now it's acting as a antigen presenting cell. Okay? It's still a B cell. It's just acting like as a presenting cell because what it's doing, it's presenting the antigen to the T helper. The only reason that this communication can happen is because T helper cells have what? CD4 receptors, which are specific for class 2 MHCs, right? Which is on the cell surface here. Okay. So now that class 2 MHC has something different on it, doesn't it? Otherwise, it would be ignored by the T cell. You get it? All right. So when this binding happens, that stimulates other chemicals to come out of the T helper, right? And this is where you get the full response from. See these chemicals coming out? Cytokines. Now it's going to trigger the cloning.
Other cytokines released from the Th2 cell, including interleukin-2, interleukin-4, interleukin-5, and interleukin-6, then bind to the appropriate receptors on the B cells, causing them to differentiate.
So we don't have to know all the different interleukins, just know that once that was bound, it triggers the T cell to start activating all the cloning on the B cell. These are the plasma cells. Now it's secreting what? Antibodies. Perfect. Okay. So that's what I wanted you to see out of this video.
So the B cells not clone until the T helper cells tell them?
Right. Right. That's why T helpers are, who said that? That's why T helpers are essential. That's why AIDS kills your immune system. Because AIDS kills off the T helper cell. So then you don't get a full immune system. So any old thing, like common cold, the person can have a cold for months and turn into a horrible respiratory infection because their immunity is just kind of shot. So they have to be on so many medications. Yeah, so, you know, cytotoxic T cells get all the glory because they can just kill outright, but without T helper, your immune system is basically nothing because it can't mount the full.
So you said without T helpers, you don't.
Without T helpers, you can't really mount the full antibody response. Yeah. Okay, let's switch to a different video. I hate that it's that low, man. That's really irritating. So what I'm looking forward to is just show you, these are T helper cells. This is the B cell. This is an APC, a macrophage. What's it going to do? Gobble up an antigen. This macrophage is acting as an APC. Once it brings it in, what's it going to do? Digest it, right? This is just showing you that it's on the inside of the cell now. Lysosomes will meet it, digest it up, break it into fragments. These are the class 2 MHCs, right? They're made from the ribosome, so they're put on the ER and then assembled by the Golgi, all that thing you learn in AP1. Either way, class 2 MHCs are coming and they're going to bind to what? The fragment. This is still in the cytoplasm. So now that antigen is going to fit on that class 2 MHC, and where is it headed? To the cell membrane, like a flag, showing that something has invaded it. So the exact same thing happens on the T cell, or excuse me, on the T cell, on the T cytotoxic side, but it just won't be on a macrophage. So now we got a class 2 MHC with an antigen fragment. So what's it going to bind to? A T helper. Beautiful. There we go. There's the T helper. There's the CD4 receptor, right? Beautiful. Okay.
What happens to them? After that? What happens to it after that? Does it survive after that, or?
It can, but sometimes it's once the chemicals are released, it stimulates the macrophage to become more deadly, and sometimes it can kill itself or just kill everything inside of itself. So these are what you call co-stimulatory factors. We don't have to really learn specifically what these are, but it's not just that one binding. It's like turning that special key for a nuclear bomb. 2 people have to do it, you know? Co-stimulation factors, cell adhesion factors will occur before it starts mounting a full response. But I'm not holding you guys to know those specifically. Just know that it has to be a co-stimulation before we release all these dangerous chemicals. Okay, so that's the macrophage doing its thing. So we saw how antibodies are produced, right? Now we're seeing how on macrophage, acting as an APC, binds to the T helper. Okay. Question somewhere?
You answered it.
Okay. So these chemicals that are released are now stimulating other macrophages to become more deadly. So now they're programmed. Now they're programmed to do more of the same thing. So they went from the macrophage now to show you the same thing we already saw with the B cell. The B cell will do what? Have its immunoglobulin receptor, bind to the antigen. What does it do? Brings it in, digests it, now puts it back on the cell membrane and binds to a T helper. Okay? So the more you watch it, the more it's the same old story. I'm just going to flip over to the T cell. All right, so here is any old cell in the body. They're just going to pick a lung cell. So this is not a B cell, it's not a macrophage, it's a tissue cell. All tissue cells have what? Class 1 MHCs, right? So now this tissue cell, right, has been invaded by some virus, right? And this virus is going to be producing different proteins on the inside of the cell. So that's why they call it endogenous, because it's on the inside of the cell. Rather than being brought in, the proteins are inside already, because the virus is inside. So now these protein fragments are going to be picked up by a class 1 MHC. So this is inside a regular cell. The class 1 MHC is traveling, made by the ribosome, traveling up. It's already bound to the foreign protein from the virus that infected it. It didn't ingest the protein was made inside, okay, from the virus inside. That's why they call it endogenous, whereas the other B cell thing was the exogenous. So now, what do we have? This is a cytotoxic T cell with what kind of receptor? CD8. Beautiful. This is our infected cell with the class 1 MHC with an antigen bound to it, an antigen fragment. There's our CD8 receptor. There we go. So now what's going to happen? It's going to release chemicals now that it's bound. We got co-stimulation factors going on so we don't kill it by mistake. You know, it's just an extra security, let's say. And it'll just cause lysis of the cell. Just breaks up. Okay. So we don't have time for this next one, but I want to just show you where it is so you'll watch it. This next one right here is actually pretty nice because the guy goes through some different, it's clearer pictures that you can see. And he kind of explains it more of in a lecture type of a way. So if you want to watch that one, that'd be good too. I'll show you what's going on a little bit. The T helper, let me go back up here. This is the helper T cell right here, right? Remember the language that we did. Helper T cells communicate with what? APCs or B cells. Helper T cells are going to ignore regular body cells, aren't they? And they're going to want to do what? Look for B cells and APCs, right? That have the class 2, right? So that's what he's telling you right there. This is a infected, right? This is an antigen right here. The helper cell doesn't go to the antigen, it goes to the APC, right? That's already been infected with the antigen. So that's the difference. What does act straight on the antigen would be something like a natural killer. All right, that's where you got to kind of know what each cell does. The helper T cell goes to the APC or the B cell. The cytotoxic T cell goes for the infected body cell. Natural killer cells and general macrophages will go for the actual antigen. Does that make sense? Yeah. Okay, so it's written on the board, right? And we've talked about what happens. So now the cytotoxic T cell, the only confusing part of this, it has it binding with this. I wouldn't go for that. So just ignore him talking about the cytotoxic binding here. The cytotoxic cell is going to do what? It's going to look for an infected cell. So let me show you where that happens. Right there. This is an infected cell. So that cytotoxic cell is going to bind to that, because what kind of MHC is on this one?